Okay, so we'll start the class now. So, hi, I'm Shrija Ghosh. I've been taking your problem solving sessions. I'm a PMRF in IIT Kanpur, and I'll be taking the seventh session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goyal. So, from the past 6 weeks, we have been studying topics of chemistry, environmental chemistry. So, we have covered different numericals related to pH and kinetics, and then uh, radioactive reactions. So now from this week onwards, we will be starting the second part of the course, that is environmental microbiology. So in this week, we basically have introduction to microbiology and basic history about microbiology. So we have few questions related to those, which was related to your week 7 assignment also. So we'll see how to solve such questions, and let's start the session. So the first question in this session is, which of the following famous person was first to find out that microorganisms are responsible for wound infection and developed the method of antiseptic surgery? So the options are Robert Hooke, Paul Ehrlich, Joseph Lister, and Louis Pasteur. Anybody wants to answer? You can either write in the chat box or unmute your mic and say.
Ma'am, Paul Ehrlich.
Paul Ehrlich is not the right answer. The correct answer is Joseph Lister. So Joseph Lister actually developed this antiseptic method of surgery, which is till date it is being followed. So before before this development, actually there were a lot of problems, lot of infections that were happening during surgeries. So Joseph Lister actually pointed out that during these surgeries we need to maintain a sterile environment, an antiseptic environment. We need to use certain antiseptics to kill the germs around it, because in a wound certain kinds of microorganisms can grow and they can cause further infection. So Joseph Lister actually developed the method of antiseptic surgery in 1867. He found a way to prevent infections in the wounds during and after surgery. So during and after surgery, what he suggested to use certain antiseptics. He asked the surgeons who were doing the surgery to wipe their hands with antiseptics, to wipe the surface around the wound with antiseptics. So that those things actually helped in healing the wound faster and prevented the infections in the wound. So he was the first to apply the science of germ theory to surgery. So what is germ theory? So Louis Pasteur actually gave this germ theory, that every infection arises from a microorganism. So that germ theory is given by I think Louis Pasteur or Robert Koch, one of the two scientists. So basically Joseph Lister applied that germ theory to surgeries, that in surgeries also the infections that were happening was due to microorganisms, and we need to kill those microorganisms. So Lister's antisepsis system is the basis of modern infection control. As I told you, that till date the surgeries that are like the surgeries that take place, all the surgeries happen in an antiseptic environment. So, Joseph Lister actually used carbolic acid as an antiseptic. He used diluted carbolic acid to wipe the surfaces and then wipe the hands and other things, like to keep the surrounding of the patient aseptic. Okay. So, the other options that we saw, Robert Hooke, Paul Ehrlich and Louis Pasteur, what are their contribution? So, anybody knows what Robert Hooke did? It's very famous actually in microbiology. Robert Hooke is the name of Robert Hooke. It's very well renowned. So,
Ma'am discovered the first micro.
yes, first cell. So, Robert Hooke first actually observed the cells. So, first observation of cells, the description that okay, life exists inside a cell. So, that was given by Robert Hooke in 1665. So, way back. Okay. After Robert Hooke, the second option was Louis Pasteur. Louis Pasteur actually gave a lot, like he had done a lot of different uh, he had made a lot of different discoveries and developed a lot of techniques. So one of the thing that he developed was discovered was fermentation. So fermentation process was explained by Louis Pasteur. Then pasteurization, it's a very famous process for disinfection. So pasteurization is done. If you would have heard your milk packets, those are always, you would have seen the term pasteurized milk. So those milk packets actually, before packaging of milk, the milk is heated at a very high temperature to kill all the microorganisms in the milk that can cause any infection. So that was also given by Louis Pasteur. Then he also disproved the spontaneous generation. And then the third option, Paul Ehrlich actually was responsible for the first effective treatment of syphilis. So he was actually screening multiple drug compounds, and he was trying to find the cure of sleeping sickness. So, but one of his drug candidate, which was later named as salvarsan, so it's a very common name, like in terms of history of microbiology. Paul Ehrlich gave this name salvarsan to one of the drug candidate, which actually was able to kill the microorganism that was causing syphilis. So he had done many animal testing in mice model and rabbits. So after that, this drug was released. So Paul Ehrlich is responsible for finding an effective treatment for syphilis. Okay. So we'll move to the next question. Question number 2. Just a second. So question number 2. In which year was the influenza virus first isolated from human sample? So from a human sample, influenza virus was isolated. So it was isolated in which year? 1937, 1933, 1957 and 1918. So the correct answer for this question is 1933. In 1933, the human influenza virus, it's a human virus. So as if you would have seen the lecture videos, the virus particles basically they cannot survive on their own. They need a host to replicate and like increase their number, increase their population. Okay, to survive they need a host organism. So influenza is a type of virus that uses humans as host. So all the common cold you would have seen, most of such uh, diseases are caused by influenza virus. So infection of influenza virus in your body causes flu kind of symptoms. So you must have heard of swine flu, H1N1. So those were all types of influenza viruses. So first time somebody isolated a influenza virus from a human sample and proving that influenza is a human virus, that was in 1933. Okay. So moving to the next question. The defining characteristic of living beings are: A. They reproduce. B. They move in response to external stimuli. C. They can grow and repair their own tissues. And D. All of the above. Anybody wants to answer?
All of the above.
Yes, all of the above is the correct answer, because all the characters that are characteristics features that are given, they reproduce. All living beings can reproduce, they can multiply, they can increase in number by reproduction. Second option, they move in response to external stimuli. So all the living cells, all the living organisms, they move in response to external stimuli. External stimuli means some external response. So they can move, they can respond to light. So you would have seen plants and trees can grow in response to light. They can use light for photosynthesis. We humans also respond to other kinds of stimuli, all the external factors that are present. All the organisms can actually respond to such factors. Then third option is they can grow and repair their own tissues. So all living organisms, being microorganism or animals, humans, plant, any organism, all the living cells basically, they can grow and they can repair their own tissues. All the cells have a repairing mechanism. So something wrong happens, the cells themselves have evolved such repair mechanisms to repair themselves. Okay, so all of the above is the correct answer, because all the features that are given are characteristics of living beings. So there are certain other characteristics that are not mentioned in these options. We will cover those also. So characteristics of a living beings. First is metabolism. So all the living cells, they can utilize energy, they can take nutrients from the environment and transform it into biomass and energy, and the output is waste excreted into the environment. So all the organisms that you can see around yourself, including your own self, including humans, you can see all the cells can take nutrients. Even if a bacteria is growing, it can take up nutrients from the environment, from the media, wherever it is growing. Okay, and it can transform those nutrients into biomass and energy for its own survival. Okay, we humans, we take nutrients from food, we eat food, we get all the food sources from environment, and then we transform it into biomass and energy, and that's how we function, right? and whatever output, whatever waste is there, it gets secreted out of our body and it goes into the environment again. So it's a cycle. We take from the environment and we give the uh, sources back to the environment. So that is first characteristic. Then second characteristic, reproduction, we have already covered in the question. So cells reproduce themselves based on their genetic code. Okay. Third is differentiation. So cells can adapt to new environmental conditions such as changing into spores to survive in hostile conditions. What do you mean by hostile conditions? So hostile conditions means some unfavorable situation. So let's say there is a cell that is growing and suddenly the pH around the cell changed drastically, it decreased or it increased drastically. So what happens in such situations, the these cells, they convert themselves into some dormant spores. Dormant means they are not growing at that stage. So when the cells were active, they were continuously growing, reproducing, metabolizing. But now what the cell has done to survive in such situation, now in that high pH or any hostile condition, it knows that it cannot reproduce or it cannot metabolize. So in that state, what it does, it changes itself into some kind of spore which is dormant in nature, which is not growing, so that it can just stay there, and once the conditions are again back to normal, the spore will again germinate into the cell and the cell will grow normally. Okay. Then next is communication. So all the cells, they can communicate with each other using some signal molecules. Okay. So in bacteria, this communication has a very interesting term given, quorum sensing. So some bacteria actually releases certain chemical compounds which are sensed by the other bacterial cells around that particular bacteria, and they take that chemical signal, like that chemical molecule, as a signal, and they respond to that. Like basically they, how humans are talking to each other, similarly those bacterial cells, those single cells unicellular organism, they are also talking to each other by sending certain signals. Okay. So communication is there in all types of cells. Okay. Then movement, cells can move in response to light, chemicals, I have already explained. And then evolution. So all the living cells are capable of undergoing they are continuously evolving actually. So cells, what happen if you keep a particular kind of cell in certain environment, and then slowly you are changing that environment. So what the cell will do, since the cell wants to survive, cell will start adapting to a new environment. It's like if you are living in your house and then after few years you are shifting to a new house, or you are going to college, to a hostel or some place. So initially you will find a change, but after a point when you start living there, you will start adapting to the new conditions, and after a point you will consider that new place as your home. So it's the same thing. So all the cells are capable of evolving. So that is a very defining characteristic of living cell. Okay. Non-living things do not evolve. You have a you have a pen, let's say, the pen will have the same characteristics in all the environments you take. Okay. So these are the characteristics of living cells. Okay. Any doubt? If you have any doubt, you can ask in the chat box, or you can unmute your mic. Okay, so moving to the next question. Question number 4. Who explained the cause and effect relationship between a pathogen and a disease? First option is Edward Jenner, second is Vinogradsky, third is Van Leeuwenhoek, and fourth is Robert Koch. So, who explained the cause and effect relationship between a pathogen and a disease? Anybody wants to answer? You can guess the answer.
Robert Koch.
Yes, Robert Koch is the correct answer. So, Robert Koch actually what he did, he gave four postulates. So, basically, he gave four, you can say, theory, or not exactly theory, postulates are not exactly theories. So, he explained the relationship between the pathogen and a disease, that a pathogen will be present when there will be a disease, and if there is a disease there will be a pathogen. So that kind of relationship, like if you have a diseased human and a healthy human, so that diseased human will have the causative pathogen, and that causative pathogen will not be present in the healthy human. So those were that this phenomena was explained by Robert Koch. So these are the four postulates, Koch's four postulates. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organism. Okay, so if there is a microorganism that is causing disease, so it will be there in the diseased person, but it won't be there in the healthy person. Okay, then second is microorganism must be isolated. The microorganism must be isolated from a diseased organism and grown in pure culture. So that microorganism, now if the microorganism is present in the diseased person, so it should be able to, like, we should be able to isolate it from the diseased organism. Okay, so let's say a person is infected with streptococcus pneumonia. Okay, it's a pathogen that causes pneumonia. So that streptococcus pneumoniae, that will be present in the person who has pneumonia, and it won't be present in the person who does not have pneumonia. Okay, and the person who has pneumonia, we should be able to get that streptococcus pneumonia, that bacteria, out of that person and culture it in as a pure culture. Okay, the third postulate is the cultured microorganism should cause disease when introduced into a healthy organism. So now once I have isolated that streptococcus pneumonia from the healthy from the person who had pneumonia, if I take the bacteria and put it in a healthy organism, that healthy organism should catch the disease. Okay, should get pneumonia from that bacteria. And the fourth postulate he gave was microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. So now I have taken the bacteria from the diseased person and grown it in a pure culture, and then that cultured microorganism I have added it to the healthy human. So from that healthy human, now that healthy human should catch pneumonia first of all, and after that person has catched pneumonia, when I'll isolate the microorganism from that diseased individual whom I have given the disease, actually I have inoculated the streptococcus pneumonia in that person. So now when I am isolating the sample from that person, I should get the original streptococcus pneumonia that I have added in that person. Okay, so these were the four Koch postulates that were given by Robert Koch. Okay, this is clear.
Yes, ma'am.
Okay. So we'll move to the next question. Question number 5. Prokaryotes can be found in the following two domains: bacteria and protista, archaea and eukarya, bacteria and archaea, and the last option is eukarya and monera. So, what will be the correct answer for this? Prokaryotes. What are the two domains under prokaryotes? Anybody? Prokaryotes have two domains. What are those two domains? Bacteria and protista, archaea and eukarya, bacteria and archaea, and eukarya and monera.
Ma'am, D, Eukarya and Monera.
Uh, it's not the correct answer. So, the correct answer for this is bacteria and archaea. So, prokaryotes and eukaryotes, there are two terms, prokaryotes and eukaryotes. Okay. So, uh, most of the all the prokaryotes actually are unicellular, and then eukaryotes, they can be unicellular or multicellular. The division was done based on the presence and absence of membrane bound organelles. Okay. So, as you would have studied, in humans we have so many organs and organelles inside our cell. So each cell has a mitochondria, you must have studied. They have a Golgi apparatus. So all these small organelles, they are present inside the cell, and all these organelles have their own cell membrane. Okay. But in case of prokaryotes, they don't have any defined organelles. So it's just one cell, one membrane bound cell, and then inside that cell every protein is just present like that. There is no further membrane division inside the cell. Okay, so those are prokaryotes. Now those prokaryotes have two domains, bacteria and archaea. So bacteria, or sometimes it's called eubacteria. So eubacteria and archaea have archaebacteria. So I'll show you the classification chart. Sorry. So living organisms are divided into prokaryotes and eukaryotes. Okay. So prokaryotes, I've told you, they are mostly unicellular and they don't have any membrane bound organelles. Whereas eukaryotes, they have membrane bound organelles. So prokaryotes don't have nucleus. Eukaryotes have nucleus in them. Okay. Now this prokaryotes, they are divided into archaea and bacteria. So archaea have archaebacteria and bacteria have eubacteria. So between archaebacteria, prokaryotes are the ones that do not have nucleus or any membrane bound organelles, whereas eukaryotes, they have nucleus and membrane bound organelles. So among eukaryotes, you can have different cellular organization, either it can be unicellular or multicellular. So all the kingdoms that have been described by Whittaker, so there are 5 kingdoms you must have studied in school. So Monera, Protista, Fungi, Plantae and Animalia. So Monera are the prokaryotes. All other kingdoms, they come under eukaryotes. So Protista, Fungi, Plantae and Animalia, they come under eukaryotes. So among that, Protista mostly unicellular, all Protista are unicellular. Fungi, some fungi are unicellular. Plantae and Animalia, most of them are multicellular. Okay, fungi, most of the fungi, Plantae and Animalia are multicellular. There are some fungi like yeast that are unicellular, and then algae comes under the Plantae kingdom, but then it is unicellular. Protozoa are also unicellular. So this is how the living organisms are classified. So under prokaryotes and eukaryotes. So you have prokaryotes that are unicellular, but they do not have any nucleus or membrane bound organelles. Eukaryotes have membrane bound organelles. So that's why they are classified differently, and basically they have nucleus and they have both cellular organization, unicellular and multicellular organisms. Okay. So moving to the next question, question number 6. Overall shape of a bacteria is determined by which of the following? Cell wall, cytoskeleton, nucleoid and cell surface membrane. So which of the following structure which of the following component determine the cell wall bacterial structure? The answer is cell wall. So cell wall determines the shape of the bacteria, the structure of the bacteria, whether it is going to be round, spherical, rod shaped, whatever it is. So how does the cell wall determine the shape of a bacteria? Let's see. So this is a bacterial cell. So as I've told you, it does not have any membrane bound organelles. So it has a cell wall, and then inside the cell wall cytoplasm is present, and in that cytoplasm all the DNA and ribosomes, they are floating. So they don't have any compartments inside the cell. Okay. So that this cell wall, it encloses the whole cell and it determines the shape. So this cell wall, based on the structure of cell wall, the bacterial cell is divided into two types, gram positive and gram negative. So gram positive bacteria, so this positive and negative don't get confused by the charge of the cell wall or cell surface. This name was given by depending on the type of gram stain they can take up. So gram positive bacteria, gram staining is a procedure of staining the bacterial cells. So gram positive bacteria can take up the crystal violet, that's why they are called gram positive, and gram negative can lose the crystal violet, so they are called gram negative. The reason of this taking up this dye is because these gram positive bacteria, they have a thick peptidoglycan layer. Now this peptidoglycan is basically the major component of the bacterial cell wall. Okay. So this peptidoglycan, it is the major component of the bacterial cell wall, and these are made of different sugar molecules, N-acetyl glucosamine, that is NAG, and N-acetyl muramic acid, that is NAM. And these sugar molecules, they are present in alternating units, and multiple molecules are present of NAM and NAG, and they form a polysaccharide, and from these polysaccharides tetrapeptide chain is attached. So amino acids, 4 amino acids are attached forming a tetrapeptide. So L-alanine, D-glutamic acid, L-lysine and D-alanine is present. Okay. So this is the structure of the peptidoglycan layer that is present in the bacterial cell wall, and this peptidoglycan layer is thick in case of gram positive bacteria, and in case of gram negative bacteria the peptidoglycan layer is thin and it is surrounded by a outer lipid membrane. Okay, so gram negative bacteria have a outer membrane apart from the peptidoglycan. Okay, so this is the structure of a bacterial cell wall, and this determines the shape of the bacteria. So there are different shapes of bacteria present. So there are bacteria that have a spherical shape, cocci, generally named as cocci. There are bacteria that are rod shaped, generally named as bacilli or bacillus. There are bacteria that has comma. Yes, so we'll continue. Sorry for that. So this is clear, I hope, bacterial structure. We'll move to the next question. Question
Clear, ma'am.
number.
Clear, ma'am.
Yeah. You have any question?
No, no, ma'am.
Yeah, okay. So question number 7. What is the genome size in a what is the genome size of a yeast? So in million bases, like how many million bases of genome size is there for yeast? So yeast is a fungus, I have already told you. So option number A is 12.1 million, B is 4.6 million, C is 137 million, and D is 97 million. So anybody wants to answer? What is the genome size of a yeast? How many million bases?
12.
12 million bases. So yes, the genome size of yeast is 12.1 million bases. Okay. So you should have an idea of the genome size of most of the model organisms. So yeast and many other organisms, they are used as a model organisms in the lab. So you should know the size of the genome size of these organisms and their comparison with human genome size. So human, or the scientific name Homo sapiens, the genome size of human is 3.2 billion. So 3.2 into 10 raised to the power of 9 bases. Okay, so it's a huge genome, and almost 30 to 40,000 genes are present in human genome. Okay, 30,000 to 40,000 genes are present, and 3.2 billion is the genome size. Whereas a laboratory mouse, M. musculus, it's a laboratory mouse. It's mostly, if you would have seen in any experiment or in any papers, it's written like these experiments were performed in a mice model or in a mouse system. So mostly this M. musculus, this species is used, and the genome size of this mouse is 2.6 billion, and number of genes is almost equivalent to that of human. So it is also 30,000. The number of genes is 30,000 for this mouse. Then there is a plant which is commonly known as mustard weed. Mustard weed, the scientific name is Arabidopsis thaliana. So this plant, the genome size is 100 million. So from billion we have shifted to million now. So 1 million is 10 raised to the power of 6. So 100 million means 100 into 10 raised to the power of 6 bases. And in this 100 million bases also, if you see, 25,000 genes are present. Now you can see the difference. Human genome size is 3.2 billion, but in that also 30,000 genes are present, and Arabidopsis has 100 million genome size and still 25,000 genes are present. So in human genomes there are a lot of repetitive sequences that are present, which does not constitute to any gene. So that's why the genome size is so long for humans, and others smaller organisms, the genome size is smaller, but the number of genes are almost similar, or it's still higher. The ratio of genome size to genes is higher for those organisms. So again, further smaller organism, So Arabidopsis thaliana was a plant. So then smaller than that is a roundworm, which is also known as C. elegans, Caenorhabditis elegans. So it's a roundworm. The genome size for this is 97 million and the number of genes present is approximately 19,000. Then further smaller organism, Drosophila melanogaster is the scientific name for a fruit fly. So you would have seen in your home, if you would have bought banana and kept it for a long time, there are small flies that fly over the fruit. So you would have seen it's very small fly, brown colored fly you would have seen over a rotten banana. So those flies are mostly Drosophila melanogaster. Those flies are mostly Drosophila species. So those are commonly called fruit flies. And they have genome size of 137 million and number of genes is 13,000. So these are also very common model organisms used in lab studies. These are used for different kind of genetic studies. Then smaller than that is yeast. Yeast is a fungus, I've told you. Saccharomyces cerevisiae is a common species that is used in labs. So the genome size is 12.1 million. Now the genome size has drastically reduced if you see, and the number of genes is also very few. Only 6,000 genes are present. In humans, 30,000 genes are present. So you can see the difference now. And yeast is a micro organism. So you can see yeast colonies with your eyes. You can't see a single yeast cell. Then bacteria, different bacterium are used. Most common bacteria used in lab are E. coli, Escherichia coli. So E. coli are very commonly used lab bacteria. These are the genome size for this bacteria is 4.6 million and the number of genes is 3200. So almost half of yeast, if you see. Yeast had 6,000 genes and E. coli has almost 3,000 genes. So almost half of yeast. And the genome size, if you see, the genome size for yeast is 12 million, but for bacteria it is 4.6 million. So almost 3 times increase in the genome size, but 2 times increase in the number of genes. So see, the smaller the organism, the lesser the smaller the organism, the higher the ratio of genome size to genes is there. Okay, the higher you will go in this organism list, you will see that most of the sequences are not a part of any genes. Those are mostly regulatory sequences and different kinds of such sequences. So then after bacteria comes the virus. So HIV, it's a very common virus, human immunodeficiency virus. It's a causal organism for AIDS, and the genome size, you see, it's very small. It's only 9,700 bases, means 9.7 kilobases, and the number of genes is only 9. Only 9 genes are present in this virus. Why only 9 genes are present in this virus and still the virus is surviving? So as I've told you, viruses survive inside a host organism, and what they do is they use the host machinery for its survival. So HIV, they have only 9 genes for themselves, but still all the replication and all the functions is happening for the virus. That's how the virus is dividing and causing infection in so many individuals, right? So what the virus is doing, virus is using the host's own replication mechanism, and it is giving just few inputs of its own, and surviving very happily without taking any efforts. Okay. And coronavirus, you have seen so many news, we all have survived through pandemics. So this coronavirus, the genome size is 27,000 to 34,000 bases. Okay. So these are some common organisms that are mostly you would come across in different kinds of lab studies. So all these organisms have a very different genome sizes and the number of genes. So you should have an idea, even if you don't mug these numbers up, but then you can just have an idea, approximate idea of how many genes are present and what is the genome size. Okay.
Ma'am, one query.
Yeah.
What is the exact meaning of like the gene? Like they are the segment of DNA or like that code one protein and another RNA molecule.
So
Means I doesn't getting.
yeah, I'll tell you.
Yes.
So genes are basically, they are segments of DNA, you can say, that have certain information and that can get translated into protein. So every individual cell has a DNA molecule. Okay. That DNA contains a lot of sequences that are called genes, like some part of the DNA can be one gene and then some other part can be second gene, and like that one organism, like for HIV I'll take it since it's the smallest in this list. So the size of the genome is 9700 bases. So the whole DNA that is in this case HIV has RNA. So the whole genetic material that is there, it has 9700 bases. So all that ATGC that is present in the HIV, it is the total number is 9700. Okay. Out of those 9700, let's say one gene is encoded by some 100 base pairs. Okay. One gene is encoded by some other 1000 base pairs. So those 100 base pairs and then the second 1000 base pairs, those are different genes. Those genes can get converted into proteins, functional proteins or non-functional, like they will have some or the other role. Either it will be a structural protein where the protein will form the structure of the virus, the coat protein, the capsid, the like whatever proteins are present in the viral particle, maybe one or two of the genes from these 9 genes are coding for those proteins. So those are called structural proteins. Then you have some non-structural proteins, they have some other function. So like for HIV, one of the very common protein that is being formed is reverse transcriptase. So it's an enzyme actually. Since this virus has RNA as the genetic material, it helps in its replication, it helps in its transcription. So so these kinds of protein, these are encoded by these genes. So from that 9700 bases, it's not necessary that all the bases are involved in some or the other genes. Okay. There can be few bases left out that are not coding for any gene, and that you can see from the genome size of humans only. 3.2 billion bases are there. That means 3.2 into 10 raised to the power of 9 bases are there. If you keep on writing ATGC continuously, you will have to write it till 3.2 into 10 raised to the power of 9 times, then you will get the human genome size. So you can think how big it is. But the number of genes is only 30,000. So do you think all the bases are involved in the genes? It's not. And if you see in mouse, it is 2.6 billion. So it is less than humans, and still the number of genes is 30,000. So that means there are more number of sequences that are not a part of genes in humans than in mouse. Okay. So genes are basically part of the genome. Okay, so all genes can be part of the like all genes, all the bases in a gene will be part of the genome, but all the bases in a genome will not be part of a gene. Okay, this point is clear.
No, ma'am, read the last line. What you say? All the gene is the
All
part
the
of the.
yeah, all the bases in the genes. So genes are also comprised of bases. Okay, one gene can be 1,000 bases, one gene can be 2,000 bases, like that. So all the bases in any gene, they will be part of the genome, but all the bases in genome is not necessarily a part of the gene. Okay, is it clear now? All genes are
Yes,
present
ma'am.
inside the genome, but all the bases in the genome, they are not involved in genes. Okay, think of a genome as a big huge string and genes as small part of that string. Okay,
Ma'am, for exam point of view, so you have to be learn the both
you
like
don't
this
have
both
to,
number.
yeah, you can like you don't have to learn the exact number, but then you should have an idea of how many genes are present in which organism, like approximate idea.
Okay, okay, fine.
Okay,
Sure.
so moving to the next question, question number 8. The flow of genetic material in microbial cells. The flow of genetic material in microbial cells usually take place from RNA through DNA to proteins. B is DNA through RNA to proteins. C is proteins through RNA to DNA, and D is none of these. So how does a flow of genetic material in microbial cell takes place? So it starts from DNA, then it goes to RNA, and then it goes to protein. So DNA through RNA to protein. So what I was explaining right now, the genes. So genes are basically, if it's the genome is made of DNA, then the genes will be part of the DNA. That DNA will be converted into RNA. That gene will get converted into RNA, and then that RNA will get converted into protein. That is how the genetic material flows. Okay. So information, whatever information, like that is mainly the gene. Every gene carries some information. Like one gene is responsible for making some enzyme, one gene is responsible for making some structure, like that. Okay, so whatever information is there, that DNA, it is stored in the DNA, it is processed and replicated. So that process replication happens. Basically the multiple copies of that DNA is formed. Okay, so genetic information in the form of DNA is copied into daughter cells. Then in the daughter cells, now they have gotten one copy from the parent. Now that DNA will be transcribed. So that DNA will serve as a template and RNA will synthesized. So complementary to the DNA, RNA will be synthesized. This is called transcription. Then from that RNA, translation will happen, so that each RNA molecule will be converted into a protein molecule. Okay, so RNA in turn serves as a template for producing specific proteins in the process of translation. Okay, this is how the genetic material flows from DNA. DNA replicates to form multiple copies of DNA, then from DNA goes to RNA by transcription, then from RNA goes to protein through translation. Okay, so it is the it is actually called a central dogma. Okay. So moving to the next question. Property of a codon for always coding a specific amino acid is called. So each codon coding for a specific amino acid, what is that property called? Degenerate, non-overlapping, non-ambiguous, and none of these. Anybody? What is this property called? It's non-ambiguous property. Okay, so the single codon, it's always codes for the same amino acid. It's not ambiguous that one time this codon is coding for some other amino acid and the second time the codon is coding for some different amino acid. It doesn't happen like that. Okay, so single codon always codes for the same amino acid, and that this describes the non-ambiguous nature of the genetic code. So there are other properties of the codon that you should know. So first is codon is a triplet. So this was described that each codon, it is given by a triplet. So AUG codes for methionine, like that each codon is a triplet. Then codon is degenerate. What do you mean by degenerate? Degenerate means there can be multiple codons that can code for same amino acid. So one amino acid can be coded by multiple codon, but one codon will code for only single amino acid. Okay. Then code is non-overlapping. None of the codes are
Please repeat the last sentence.
Degenerate. So degenerate means, I'll
Yes,
explain in the next slide actually what is degenerate. So one codon, let's say AUG, AUG will always code for methionine. Okay, methionine is an amino acid. AUG will code for methionine always, but there is a chance that there is some amino acid like lysine or valine, they can be coded by 2 or 3 codons simultaneously. Okay, so that is called degenerate property of codon. Then the code is comma-less, so there is no gaps or comma between the codes. The codes are read continuously. So you read in frame of 3. So you will read the first 3 codon, make an amino acid, then next 3 codon, make an amino acid. There is no gap or comma between those codons. Then code is unambiguous. I have already explained, like each code codes for single amino acid, and code is universal. So AUG will code for methionine in bacteria and in human. It is universal in all the organisms. Okay. So the next question is actually, genetic code is called a degenerate code because. So now this will clear your doubt. So a genetic code is called a degenerate code because the options are, there are 64 codons for 20 amino acid. One codon has many amino acids. Each codon codes for only one amino acid. More than one codon codes for a single amino acid. Okay. So the answer for this question is, yeah.
Ma'am, answer is A.
Yeah, answer is, there are actually more than one correct answer for this question. If you read the options carefully, the code is degenerate, yes, because there are 64 codons and 20 amino acids. So one codon can code, one amino acid can be coded by 2 or 3 codons simultaneously. Okay. One codon has many amino acid. This is a wrong statement. One codon has one amino acid. We have already seen that it is non-ambiguous. Each codon codes for only one amino acid. This is also a true statement. So genetic code is degenerate because each codon codes for one amino acid. So since each codon is coding for one amino acid, one amino acid can be coded by 2-3 codons, but one codon cannot code for 2-3 amino acids. Okay, and more than one codon codes for a single amino acid. This is also true because that is why it is degenerate, right? More than one codon codes for a single amino acid. So the answer for this question is all the three options. There are 64 possible combinations of 3-letter nucleotide sequences that can be made from the 4 nucleotides. Of these 64 codons, 61 are used to code 20 amino acids and 3 are stop signals. Although each codon is specific for only one amino acid, the genetic code is described as degenerate or redundant because a single amino acid may be coded for by more than one codon. Okay. So moving to the next question. You understood what is degeneracy of codons, right? Why a codon is degenerate.
Ma'am, is it possible? Can you explain little concept of the what is gene? What is code? Like what is the role of they can what they can play role of the code?
Okay. So you know about different bases, right? A, T, G, C, they make up the genome of an organism. You know that part, right?
Yes, ma'am. But
Yeah. So once a genome is converted into RNA, so difference between DNA and RNA in this context is the DNA has a thymine base, and in place of thymine, in place of T, there is a U present in RNA. Okay. So in DNA it will be ATG, but in RNA it will be AUG. Okay. So this is the difference in this term. So when there will be a DNA molecule, from that RNA will form. So by complementary base pairing, different RNA molecules will form. So let's say I have a DNA molecule with a sequence T TAC. Okay, I have a DNA molecule with a sequence TAC. Okay, so T base pairs with A. So in when RNA will be formed on this DNA template, the first base that will be entered will be A. Then in front of A, the second base that will be entered will be U. I told you because A pairs with T, but in place of T we will put U. So AU, and then in front of C, the base that will be added in the RNA will be G. So you have the first three bases as AUG. Okay. Now this AUG in total, so once the RNA is transcribed, now from this RNA, three bases together forms a code. Okay. So this AUG is one code. Then CUA is another code. Like that, 64 different codes are present. Okay. By permutation combination, you will see 64 different codes are present. Now from those 64 codes, 61 codons codes for 20 amino acids. So different 3-letter codons, they code for different amino acids. Okay. And then like that, so from one RNA molecule, so from one RNA molecule, there will be it will be divided into three bases, and then each 3 bases will code for one amino acid. So like that you will get a amino acid chain. Now what is a amino acid chain? They will ultimately make a protein. That amino acid chain is the primary sequence of a protein. That protein will get folded and then function inside the cell. So that is the process how a cell makes its own protein and survives. Okay, is that clear?
Yes, ma'am.
Yeah. So I'll see in
Okay,
the
fine,
next.
madam. Thank you.
Yeah, I'll see in the next class. I'll add some more slides related to that if necessary. Okay. So question number 11 is studies of similarities and differences among the genomes of multiple organisms is carried out in. Option number A is proteomics, option number B is functional genomics, C is structural genomics, and D is comparative genomics. So the answer for this question will be studies of similarities and differences among the genomes of multiple organisms. So we are studying different organisms and comparing. So the answer will be comparative genomics. Okay, we are comparing, we are seeing the similarities and we are seeing the differences. So we are basically comparing the genome of two organisms. So it is comparative genomics. Okay. The other options, proteomics, functional genomics, and structural genomics. What are those? So proteomics is basically study of proteins, you can say proteome. So proteome is the set of proteins produced in an organism or in a system, and study of those protein, the whole proteome, it is called proteomics. Then you have functional genomics and structural genomics. So functional genomics, it focuses on the dynamic expression of a gene product in a specific context. So that means we are studying one function of the genome. Okay. So let's say specific developmental stage. So we are studying that developmental stage. So we will study how that genome is functioning in an organism or during a disease, like that. Okay. Structural genomics, it's the study about structures. So the genome is coding for different types of proteins. Every protein or every macromolecule in that matter has a specific structure. So study of those structure is structural genomics. Okay. So the next question, question number 12. Which of the following may use RNA as its genome? A bacterium, an archaeon, a virus, and a eukaryote. So I have actually told you in the previous slides the answer for this question. Do you remember which has a RNA genome? Which organism has a RNA molecule as a genome? Yes, you can answer.
Virus.
Yes, I have told you na. HIV is actually an example of a RNA containing virus. There are viruses that contain DNA also, but then there are certain viruses that have RNA. So coronavirus also is a RNA containing virus. Okay. So since it's a recent example, you all should know this part. So the only one here that may have an RNA genome is a virus. Virus may have DNA or RNA as their genetic material. All other organisms have DNA as their genetic material. Okay. So moving on. What is the type of extremophile that thrives in areas with a very little water where it is really dry? So in a very dry environment, which extremophile can survive? So the options are halophile, thermophile, xerophile, and radioresistant. Anybody knows? So the answer for this, yeah.
Xerophils.
Yes, xerophiles. So the answer for this question is xerophiles. So xerophiles basically they can survive in these dry areas. So xerophytes are actually xerophytes is a term used for the plants that can grow in dry area, and xerophiles is the term used for bacteria that can grow. So other extremophiles that we have are halophile, thermophile and radiophile. So halophiles are basically organisms that can grow in extreme salt concentration. So very high salt concentration they can grow and tolerate. Thermophiles, they can grow and tolerate at high temperatures. And radio resistant means they are resistant to radiations. So different types of radiations we have studied in the radioactivity chapters, right? So in that, there are certain microorganisms, they can actually survive radiation. Okay. So the next question is, what is the type of extremophile that lives in a very salty environment such as the Great Salt Lake? So I have already told you the answer in the previous set. Halophile can survive in very high salt concentration. Okay. So there are certain, sea water is salty, right? So sea or ocean water. So how do you think if you try to isolate microorganisms from sea water, you will easily get many colonies. So those organisms, they are surviving because they have a capability of tolerating that high salt concentration. So they are halophiles basically. Okay, not all bacteria can survive in high salt concentration. If you take a bacteria from, let's say, a gut and then try to culture it at a high salt concentration, they might not survive. Okay, but since you know that your stomach has a very low pH, okay, so if you isolate a bacteria from your stomach and then try to grow it at a very low or very acidic condition, it will grow. So those are acidophiles, they can grow in acid nicely. So there are different, the term phile means loving. So halophile means halo loving or salt loving. Thermophiles means thermo is related to temperature, so high temperature loving. Xerophiles, xerophytic conditions means dry conditions, so dry condition loving. Radiophile, radiation loving. Okay. So halophiles are a group of organisms with the ability to prosper in hypersaline conditions. In this kind of extreme environment, the salt concentrations are in excess of seawater and can reach 20% to 30% NaCl. Halophiles require high salt concentration to thrive in these unusual habitats. Okay. So moving to the next question, question number 15. The binomial name of a microbe is composed of its kingdom and genus name, its genus name and order name, its family name and class name, and its genus name and species name. So binomial
Genus.
yeah, what is the answer? Genus
And species.
Genus and species, yes. So any microorganism, when it is written, you will see it is written like the first word will be written with a capital letter and the second word will be written with the small letters. The first word that is written is the genus name and the second word that is written is the species name, and both the words are written in italicized format. Okay, or when you are writing in your notebooks, we generally make an underline below the name of the genus and species to show that it is name of a organism. So this nomenclature referring to naming and this nomenclature, this binomial nomenclature system, it was introduced by Carl Linnaeus. This means that a bacterium has a name which is composed of a genus name that tells you which genus it belongs and a species epithet which together with the genus name gives a unique name to a bacterium. So like example is Moraxella bovis. Okay, so Moraxella bovis, the genus name is Moraxella, so it belongs to the Moraxella genus. Bovis, it uh, it is the species name. It indicates that the bacteria has been isolated from a cattle. So you know bovine is a term used for cattle, mainly pigs and all. The term bovine is used, right? So bovis comes from the word bovine, and it shows that this particular species is isolated from a cattle. Okay, and it is always written in italics. This is clear? So the name of the person who has given this binomial nomenclature, Carl Linnaeus, that is also very important. You should remember that. So in microbiology, there are certain like names of certain scientists you should remember, because it's very important for the history of microbiology. Okay, so Robert uh, Robert Koch, Louis Pasteur, Carl Linnaeus, Whittaker. These are the names you should see. You should go and see what these people have done, and like they have actually contributed greatly to microbiology, to the field of microbiology. So question number 16. Gram-negative bacteria that causes stomach ulcers is Helicobacter pylori, Bacillus anthracis, yeah. I hope my screen is visible, right? So, question number 17. Who isolated Streptococcus pneumonia for the first time? A. Robert Koch, B. Edward Jenner, C. Anthony von Leeuwenhoek, and D is Louis Pasteur and George Sternberg. So, who isolated Streptococcus pneumonia for the first time? Do you know the answer?
Ma'am, answer D.
Yes, option D. Louis Pasteur and George Sternberg, they have isolated Streptococcus pneumonia for the first time. Okay, so you know the contributions of the other scientists given. Robert Koch, we have already discussed. He has given the Koch postulates. He has given the germ theory also, I think. And Leeuwenhoek, he is actually called the father of microbiology. He invented the first microscope. Okay, then Edward Jenner has also contributed very significantly. He made the vaccine for smallpox. Okay, so before that the concept of vaccine was not present, so he actually made the first vaccine, and it was for smallpox. Okay, so nowadays the vaccination is very common process, and after the pandemic everybody knows about it, but Edward Jenner was the one who actually made this brought this concept into life. Okay, so moving to the next question, question number 18. A newly discovered microscopic structure is hypothesized to be a living organism. Which of the following lines of evidence would support the contention that this organism may be alive? Option A is it contains DNA, B is it is made of a single cell, C is it utilizes energy, and D is all of these. So what feature makes it a living cell? So I've discussed the characteristic of a living cell, so you can guess which answer should be correct. Should it contain DNA? Yeah,
D.
It is made of a single cell. For a cell to be alive, it should contain DNA, it should utilize energy, do you think it should be made of a single cell for it to be living?
No, it's made of the many cells.
Yeah, so option number A and C are the correct answer. Okay, it should utilize energy and it should contain DNA. Okay, so the next question, question number 19. A virus is considered to be non-living when it is outside the host because I've discussed about virus enough in this lecture, so you should be able to answer this. Option A is it cannot reproduce. Second, it cannot utilize energy. Third, it is mobile. Fourth, it is made of a single cell. Why a virus is considered non-living outside the host? Which options are correct? More than one option is correct.
Ma'am, answer A and B.
Yes, it cannot reproduce and cannot utilize energy. So it needs a host.
Ma'am, what is the meaning of C? Like it is mobile, like what is
Mobile
the
means movement. Mobile means movement, so it can move on its own. So it is not mobile also actually, viral particles. So viral particles are just like particles of protein with nucleic acid inside it. So once it is inside a host cell, then only it can reproduce and it can grow, divide, make many copies of viral particles and then infect the next host. So once it is outside the host body, it's just like a protein molecule. Okay, it doesn't have any life on its own. Okay, mobile means mobility. Mobile, the word mobile comes from mobility. So mobility means movement. Okay, so virus is considered to be non-living when it is outside the host because it cannot reproduce and utilize energy. Okay, so the next question, what is Whittaker's classification based on? So I have taken this name few times in the session, Whittaker's classification. I have told you also about the 5 kingdoms. So what is it based on? Mode of nutrition, cellular organization, presence or absence of nucleus, and all of these above. What do you think the answer is? How did Whittaker classify the organisms?
Ma'am, all of the above.
Yes. So Whittaker's classification is based on the mode of nutrition, cellular organization, and the presence or absence of nucleus. So I have explained presence or absence of nucleus. How do we divide? Prokaryotes and eukaryotes. Okay, so prokaryotes have, like, they do not have nucleus and eukaryotes have nucleus. So that is the difference. Cellular organization also I have told you. So unicellular, multicellular, based on that. Mode of nutrition, this is another thing, this I haven't explained yet. I'll explain it in the subsequent slides. So mode of nutrition, whether it is taking carbon dioxide as the carbon source or an organic compound as the carbon source. So those things also were considered when Whittaker was classifying the organisms. Okay. So these are the 5 kingdoms of classification. So Monera, Protista, fungi, plantae and animalia. So I think I said something wrong previously. Apologies for that. So Monera is basically Monera comprises of Monera and Protista comprises of all the bacteria. So I'm sorry. So Monera comprises of all the bacteria, so all the prokaryotic organisms. So both archaebacteria and eubacteria, they will come under this Monera category. Then from Protista till Animalia, all the organisms are eukaryotic. That means they have nucleus and membrane bound organelles. Okay. Now Monera and Protista are unicellular. Fungi, they can be unicellular or multicellular depending on depending on the species. So yeasts are unicellular, but other fungus like rhizopus, they are multicellular. Then Plantae and Animalia, you all know it has tissue and organ level of organ system level of cellular organization. Then mode of nutrition. So this autotrophic, heterotrophic, this I will explain in the next slide. Nuclear envelope or nucleus, so that is absent in prokaryotes and then present in all the eukaryotes. Cell wall, most of the Monera have non-cellulosic cell wall. So I have explained the bacterial cell wall structure. It has mainly peptidoglycans, so polysaccharides, sugar molecules were there, right, in the peptidoglycan, N-acetyl glucosamine and N-acetyl muramic acid. So those they multiple units of NAM and NAG were attached. So polysaccharides, polymers of saccharides, plus amino acids. Amino acids were present below below these NAM NAG units. A small tetrapeptide was present in the peptidoglycan chain, so amino acids are present. Cell walls of in Protista, some species have cell walls, not all species have cell wall. Okay, but all Monera have cell wall. All fungi have cell wall, and it is mostly made of chitin. Chitin is another molecule. Okay, all the plant cells also have cell wall. They are cellulosic cell wall. Animalia, in the animal cells, the cell wall is absent, only cell membrane is present. So all the cells inside your body, they do not have any cell wall present. Okay, that's why animal cells are more delicate to when handled in lab than the bacterial cells, because they don't have cell wall, so they are more susceptible to bursting, or they are delicate. Okay, so in the next question I will explain what are these mode of nutrition. Photoheterotrophs, have you heard of the term photoheterotrophs? What do? They get energy from light and feed on other organisms. They get energy from light and are self-feeding. They derive energy from chemicals and are self-feeding, or they derive energy from chemicals and feed on other organisms. So which option is correct? Yeah. Which option is correct?
Answer B.
B. Option number B is not actually correct. So by the term photo, you got it right. Get energy from light. That part is correct. Okay. Now we have two options, A and B, which have get energy from light. But heterotrophs, what do you mean by heterotrophs and what do you mean by autotrophs? So one is self-feeding and one is feed on other organisms. So autotrophs are the ones who are actually self-feeding. Okay, autotrophs feed on like they can make their own food source, you can say. Like in schools you would have studied about plants. So plants can do photosynthesis, they can make their own food themselves. So heterotrophs, they feed on other organisms. Like we are heterotrophs, we can we don't make our own food inside our body. We eat food from the external sources. We feed on other organisms. We either feed on plant sources, we get food from plants, or we, if you are non-vegetarian, you feed on animal sources as well. Okay, so that means heterotrophs means feed on other organisms. So photoheterotrophs means get energy from light and feed on other organisms. Okay, so the answer is A. Now I will explain different modes of nutrition. So all organisms, they can either get the so all organisms need carbon for biomass and energy, right? Everything is made up of carbon only. So your basic chemical formula would be carbon only. So carbon, oxygen, so like that. So from where will you get the carbon? You can either get the inorganic carbon from CO2, or you can get the organic carbon. So organisms that feed on inorganic carbon, that is CO2, they are called autotrophs, and organisms that feed on other organisms, other organic sources, other organic carbon sources, they are called heterotrophs. Okay, in that autotroph, now you can have two energy sources. If your energy source is light, the term photo will be added. If the energy source is chemical, some other chemical, not light, the term chemo will be added as a prefix. Okay, so in both autotrophs and heterotrophs, you see, if the energy source is light, then it is photoautotroph or photoheterotrophs. If the energy source is chemical energy, it is chemoautotroph or chemoheterotroph. Okay, then those photoautotroph, they can either use H2O, that is water, to reduce carbon dioxide, or they can use some other sources that are anoxic. So H2O means basically oxygen, so they can oxidize the, so they can basically reduce the CO2 using that H2O, using the oxygen of H2O. So that is oxygenic. Okay, aerobic, oxygenic. So photosynthesis is a oxygenic process. It plants uses H2O and they reduce CO2 for their biomass and energy. So that is a oxygenic process. Anoxygenic process, that means in absence of oxygen, also certain organisms survive. So how do they survive? So in place of H2O, they have H2S as the H2S that reduces carbon dioxide. Okay, so if they are using H2S, if they are not using H2O and using something else, that is an anoxygenic mode of respiration. And there are certain bacteria that actually uses H2S. Those are green and purple bacteria. Okay, green and purple sulfur bacteria, these are anoxygenic photosynthetic bacteria. They can make their own food source, but they don't use oxygen. They are anoxygenic. They survive anaerobically. Okay, now in chemical sources, chemoautotrophs, they are also making their own food source, but they are using chemical sources as their energy. Okay, they don't need light, so they use other chemicals like hydrogen, iron, sulfur, nitrogen. Okay, like that. Then the second kind of organisms under which we also come, organic carbon. So all the other organisms that are feeding on other organisms or other organic molecules, those are heterotrophs. Now heterotrophs also have two categories, light energy and chemical energy. So photoheterotrophs and chemoheterotrophs. Among that photoheterotrophs, you have green and purple non-sulfur bacteria. So sulfur bacteria were autotrophs, but non-sulfur bacteria, they are also using light energy, but they cannot grow by themselves. They need to feed on other organic sources for their growth. for getting the biomass and energy. Okay. Among the chemoheterotrophs, now you have again two categories. This TEA is terminal electron acceptor. Now if the terminal electron acceptor is oxygen, again oxygen means aerobic. Aerobic respiration is happening. So such organisms are categorized in one category. So under that category, all the animals, most fungi, protozoa, bacteria come. Okay, most of the organisms that you see around yourself comes under this criteria. But if there is no oxygen present, do you think life doesn't exist there? Life can still exist there without oxygen. In such cases, the terminal electron acceptor is something else. So instead of oxygen, it can be sulfur or it can be some other compound. Okay. So in such cases, it can either be an organic compound or it can be an inorganic compound. So if it is an organic compound, it is for the example is fermentative bacteria like lactobacillus. So you have seen curd, curdling of milk. You have seen people add some curd to the milk and then the next morning the whole milk turns into curd. So that is a fermentative process, and that is facilitated by certain bacteria present in the curd called lactobacilli. And if the terminal electron acceptor is not oxygen and it is an inorganic compound, it is called anaerobic respiration. Example is Clostridium. Clostridium, I have told you, Clostridium botulinum, it produces a toxin, so it's a pathogen. Okay. So this is clear, that mode of nutrition, different mode of nutrition. I hope this is clear.
Ma'am, it's clear. Ma'am, in exam point of view, we have to be learn like this, all this point of view.
So you should know what do you mean by autotrophs and heterotrophs. Okay. I hope that part is clear. Autotrophs that can make their own
Yes,
food
ma'am.
source or that can survive on their own using inorganic carbon source. Heterotrophs that need organic carbon source. Then among the autotrophs and heterotrophs, this division is common, chemo and photo. So you should understand what do you mean by photo, what do you mean by chemo. Other than that, you don't have to memorize all the further classification. You should just understand how this classification has been done. So under photoautotrophs, you can see like now it is using light as the energy source. It is using CO2 as the main carbon source. Now what is next thing? Now how will it incorporate CO2? How will it use CO2 to generate biomass? So it can use either H2O, like either oxygen or some non-oxygenic molecule. So based on that, differentiation is there. Then similarly in chemoheterotrophs also, either the terminal electron acceptor is oxygen or not oxygen. Based on that, division is there. So that is something for your understanding. But then you should definitely know what is what is autotroph, heterotroph, and what do you mean by photo and chemo. Okay, that term if you know, you will be able to answer the questions. Right. So if you see in this question that I asked now, so now you have understood all the modes of. So now if you see in this question that I asked now, you see get energy from light means photo, feed on other organisms means heterotrophs. So now you can see get energy from light and feed on other organisms, this is photoheterotroph. Now the second option, get energy from light and self-feeding. Self-feeding means autotrophs. So it will be photoautotrophs. The third option, derive energy from chemicals. So derive energy from chemicals means chemo, and self-feeding means autotrophs. So it will be chemoautotrophs. So like that you have to think, like from what is the energy source? Energy source is chemical, so chemo. What is the carbon source? Carbon source, if they are feeding on other organisms, if the carbon source is organic, it is heterotroph. So chemoheterotroph. If the carbon source is inorganic, they are self-feeding, so it is autotroph. So that's how you classify chemo-autotroph, chemo-heterotroph. Okay, photo-autotroph, photo-heterotroph. Is that clear? So this is where we end this session. So thank you all for joining.